I'm really pleased today to introduce our third candidate for the Population Community Ecology position in EEB, Dr. Julian Resasco, who is currently a postdoc at the University of Colorado, Boulder. Before that, he received his PhD in 2013 from the University of Florida. And he has done a very diverse range of work on working in particular in these fragmentation experiments, but with a lot of different organisms. There are at least one of our faculty members who is exceedingly glad that he has worked on ants, and but as well as pollinators, putting in a proposal now on beetles, and on a range of questions in community ecology. He has an excellent publication record, number of papers in ecology and ecography, in ecosphere, in tree, so has published in our major journals in ecology. And we are delighted to have him here with us today. So all yours, Julian.
Okay, thanks for the really nice introduction. Can you guys hear me okay? There were some issues with the mic. It's been great to meet several of you and see your campus, and I'm looking forward to interacting with more of you over the next day or so. We live in a heavily modified world with intense threats to biodiversity. Agricultural lands cover roughly 38% of the Earth's surface, and recognizing that agriculture and biodiversity are not necessarily always at odds, this conversion has come at large losses to biomes around the world. 70% of grasslands, 50% of savannas, and roughly a third of forests have been lost. So this is an example of habitat loss. This is the Brazilian Atlantic forest, and in this left pane here we see the original forest cover, and in the right pane we see the current forest cover. It's undergone massive habitat loss, and typically under such loss the remnant habitats tend to be fragmented. They tend to be smaller, more isolated, more affected by edge. And to get an idea of the scale of this problem, if we consider forest worldwide, 70% of that forest area is within 1 kilometer of one of these human-made edges. So this could be an edge of a field or a road. So habitat loss and fragmentation are considered to be the greatest drivers in biodiversity loss. We also have really important drivers like climate change and invasive species, depicted by this invasive ant pioneer moving across the plain. And the central goal of my research program is to better understand how changes in the environment affect biodiversity, so we can understand how to mitigate those effects. And the drivers that I'm interested in are habitat loss and fragmentation, climate change, and invasive species. And to address these questions, I use field experiments, often at very large scales, to test ecological theory and link pattern and process. This is an outline of my talk. In the first part of the talk, we'll be talking about corridors and how they might affect invasion, as well as biodiversity of native communities. In the second part of the talk, we're going to talk about fragmentation and how it can affect things like food web structure and species niches. And in the last part, I'm going to be talking about temporal variation and drivers of stability and plants, pollinators, and their interactions. So during this talk, I'll largely be focusing on fragmentation experiments. So these are the largest fragmentation experiments in the world. Among them is the Biological Dynamics of Forest Fragments project in Brazil, where Chris Dick has worked. And so these experiments are rare. There's just a handful of them. It's logistically difficult to do experiments at these scales and especially to maintain them over long time scales. But they're also extremely valuable because they allow us to test for the effects of fragmentation while controlling for variables like area, edge, non-random habitat loss. And they allow us to do this with randomization and replication, all the things that we want in experiments at scales that approximate those of management and conservation activities. I do work in 2 of these experiments, the Wog-Wog habitat fragmentation experiment in Australia and the Savannah River Site corridor experiment in South Carolina. So first, thinking about corridors. Corridor plans are of interest in conservation across vastly different spatial scales to mitigate the negative effects of fragmentation, ranging from this wildlife overpass at Banff National Park that links up these 2 parcels of forest across this highway, which might be a source of mortality or a barrier to movement for wildlife, to WildLink, which is a program here in Michigan that aims to restore and maintain connectivity among wildlands by working with private landowners in counties in the northern part of the state. To at the whole state level in the state of Florida, there's a corridor initiative to link up protected areas across the state from the Everglades up to the Okefenokee Swamp in Georgia and across the Panhandle. And there's even interest in corridors at continental scales. For example, the famous Y to Y, Yellowstone to Yukon corridor. And these scales of corridors are going to be really important as species ranges shift with climate change. But there have been and continue to be concerns about negative effects of corridors associated with them as conservation tools. And prominent among these concerns is the concerns that corridors could spread invasive species by the same principles that allow them to spread the target species that we want to spread. But despite these high-profile concerns over several decades, this is still a big question. We still know very little about this. The Savannah River Site corridor experiment in South Carolina provides a great opportunity to test theory in spatial ecology, and also to test the strength of corridors as conservation tools, and also to test for these potential negative effects that are much less often studied. This experiment was designed by Nick Haddad at NC State and Doug Levy, my PhD advisor, while he was at the University of Florida, which is where I did my PhD. And the setting for the corridor experiment is the longleaf pine ecosystem. The longleaf pine ecosystem is characterized by this open structure where you have scattered longleaf pine trees and an herbaceous understory. This structure is maintained by fires. It's a fire-adapted and a fire-dependent ecosystem. These longleaf pine savannas have some of the highest levels of biodiversity and endemism in North America, and they're habitat for rare and iconic endangered species like this red cockaded woodpecker. And the ecosystem itself is endangered. So if we consider the historical distribution of the longleaf pine ecosystem, it used to cover most of the southeast, and this is what's left of it today. Less than 3% of it remains, and what remains is highly fragmented. So I'm going to go through and talk a little bit about the experimental design of the corridor experiment. So this is one experimental landscape, or one block, and there are 8 of these blocks replicated throughout the landscape at the Savannah River Site. So we have this pine plantation matrix here in the green, and then we have these patches, a center patch and 4 peripheral patches that are cut into that pine plantation matrix. And and those peripheral patches are 150 meters away from that center patch, and there's a corridor of the same habitat, of open habitat, connecting the center patch to one of their peripheral patches. And because adding a corridor adds area, the area of the corridor is added to those peripheral patches in one of 2 ways, either to the back of the original square in this rectangular patch, or these little half corridors on the sides of these squares in these wing patches. So if we consider the area of those original squares plus the corridor, we have equivalent area in what we'll call the connected, winged, and rectangular patch. So this is what it looks like in the actual experiment when you're standing inside that center patch and you're looking down the corridor at that connected patch. You can see a productive, diverse understory there in contrast to what we have in the matrix, which is mostly dominated by just pine straw. And these patches, we're restoring these patches to longleaf pine savanna. We do this with frequent prescribed burns, planting of longleaf pine seedlings and planting of longleaf pine understory species, and mechanical removal of loblollies and hardwoods. So I mentioned that there's this big question about the concern of corridors spreading invasive species. And so the question I pose here is, do corridors lead to greater fire ant invasion? And if they do they do this to the detriment of the co-occurring native ants that they live with? So you may be asking yourself, why ants? Or some people may not be asking that, I guess. But ants are what ants and insects are what Ed Wilson calls the little things that run the world. They're abundant in most ecosystems. They're important in terms of their interactions. They perform important ecosystem functions and services. They are indicators of ecosystem health and indicators of diversity and other taxa. And they also can include among them some of the most notorious invasive species that can displace other species in their community, like this fire ant. The fire ant, Solenopsis invicta, is one of the most notorious and hated invasive species in the southern United States. You're lucky that you're too far north here to get them for now. They originated in, like myself, in South America and were introduced into the United States. They were introduced into the port of Mobile, Alabama, where they spread throughout the southern United States. And from the southern United States, they went on to invade other areas around the world. Wherever they go, they cause great ecological and economic damage. And they have these two very different social forms, monogyne and polygyne, that I've talked to some of you about, that have very different traits that can affect how they use the landscape. So I'll talk about that next. So the first is the monogyne social form. Monogyne means single queen. They contain a single egg-laying queen per colony. And they're really good dispersers. So these virgin queens fly out of their nests in their mating flights, high up in the air, and mate in the air, and descend to found independent colonies far away from their natal nest. The workers have territorial behavior. They're aggressive against non-nest mates. And this behavior leads to territories that are over-dispersed in space and relatively low densities of fire ants. I don't know how to make that thing stop. And so with these lower densities of fire ants, we should expect relatively lower impacts. Okay, and the social form can be determined by this GP9 gene, by the dominant allele. So this is a little cartoon to drive home those traits. The monogyne social form, they disperse well, they mate high up in the air, and they fly down, and they found a colony that is where the workers are territorial, so they establish at low densities. In contrast, the polygyne social form, polygyne means multiple queens. They have multiple egg-laying queens per colony. They're relatively poor dispersers, so if they have mating flights, they're low to the ground, or more commonly, they disperse on foot by budding off of their natal colony. And the workers are non-territorial against non-nest mates of other polygynes. So you can get intermixing of colonies, and so they establish it at these high densities because they don't have those discrete territories. And with those higher densities, we should expect relatively higher impacts on other ants. So a little cartoon for the polygyne, poor disperser, establishes at high densities. So we might want to add to those questions monogyne and polygyne in their response to corridors. So I collected data using pitfall traps and nest counts and collecting data directly from nests to determine social form. Found about 49 species. I've done a lot of ant collecting around the Savannah River site, and so I think the regional pool might be something around 100 species. Fire ants made up 68% of all ants, so they're major players in these patches, and they made up almost 100% of the non-native ants. So they are the major invasive species of ants in the system for now. I found that blocks were either dominated by one social form or the other. 5 of the 8 blocks were dominated by the monogyne social form. 3 of the 8 blocks were dominated by the polygyne social form. I looked for underlying differences in these blocks in terms of previous land use or orientation, and I didn't find support for that. But interestingly, 2 of these blocks were more recently created than the rest of them, and they were both polygyne blocks. So I'll come back to this idea. So to assess the effect of corridors on invasion, I'm going to present here prevalence and pitfall traps, or proportion of pitfall traps per patch that were occupied by fire ants. So first I'm going to show for the monogyne blocks, and here the monogyne queen wants to remind us that she's the one that disperses well and establishes at low densities. So if we look at prevalence in monogyne blocks, we see no difference in prevalence between connected and unconnected patches. However, when we look at polygyne blocks, we first of all see that reflecting those higher densities, we have a higher prevalence of ants, but we actually have a 36% higher prevalence in connected patches than unconnected patches. So what does this mean for, well, presumably this is because in this monogyne social form, they can disperse really well, colonize patches, regardless of whether or not there's a corridor. And in these polygyne blocks, to assess the effect on native biodiversity, I'm going to present rarefied species richness. So you're going to see the rarefied species richness with 95% confidence intervals. And so in these monogyne blocks where we didn't see any effect of corridors on fire ants, we see no effect on ant species richness. In contrast, in these polygyne blocks, we first of all see that species richness is much lower, reflecting this relationship with the higher fire ant prevalence we have, the lower species richness we have. So we see overall lower species richness, but we actually see lower species richness in those connected patches where the prevalence of fire ants was higher. So revisiting those questions, do corridors lead to greater fire ant invasion? It seems that they do in the case where we have this polygyne social form, and it is to the detriment of co-occurring species. So what are some take-homes from this part of the talk? Well, this is the first experimental evidence that corridors can help spread invasive species. It highlights the importance of phenotypic variation within a species and how that can affect how species use the landscape features like corridors differently. It suggests that species traits can help us predict which species are going to benefit from corridors and which ones aren't. So in the case of invasive species, if we have an invasive species that's a poor disperser but a good invasive species by other measures, we might expect that they might benefit from corridors. But if we consider invasive species are often really good dispersers. So they're good at they might often be good at getting around whether or not we have the corridor there to help them get places. So I co-authored a meta-analysis where we looked at the literature of potential negative effects of corridors, and including the effects on invasive species. And apart from my study, we didn't find any support of corridors spreading invasive species. So the weight of evidence still supports corridors as conservation tools, I would say. There's this cool simulation study by Hudgens and Haddad where they simulated populations under different conditions under different traits. And they found that under population traits that resembled those of the polygyne fire ants, namely fast population growth and low dispersal, populations achieved higher abundances with connected patches, but those effects were transient. So over time, that effect went away and abundances became equal between connected and unconnected patches. So this led me to do a follow-up study where I asked, are the effects of corridors on polygyne fire ants transient? So I went back and I resampled in 2014, 15, and 16. And I found that sure enough, it seems like the effect of corridors on polygyne fire ants are transient, where we had a significant effect in the original study. We no longer had a significant effect, and that effect size became smaller. But the effect for species richness, while we had a little bit of overlap in the 95% confidence intervals, the magnitude of that effect remains similar. So this suggests that there might be a legacy effect of past invasion that doesn't just snap back like that. So the data for 2015 and 16 are being processed still, and it'll be interesting to see how this relationship changes over the next few years. So lots of ongoing and future research in the corridor experiment. The work I just presented illustrates how important having these long-term studies are for understanding the dynamics of what's going on out there. So together with this team of PIs, we're monitoring community dynamics over a decade as part of an NSF LTREB. So there's going to be research coming out of this for years to come. We have a very cool website called conservationcorridor.org that aims to link the science and practice of corridors, where I contribute as writing accessible digests of new science papers on corridors and connectivity. Also want to mention that this project is a great environment and a great community for students to work and learn and receive mentoring. For example, I participate by mentoring REU students. I had an REU student last summer, Deaja Sanders, who did a great project on seed predation by harvester ants. And I'm putting in a proposal to co-mentor one or two REUs this coming summer. And finally, I'm in the process of designing a study for a proposal with Rob Fletcher and Jim Austin of the University of Florida. I'm excited about these ideas. I think they could influence basic and applied connectivity research. So I'm going to spend a few minutes talking about this system where we're looking at connectivity at different scales and how that can affect genetic structure and recolonization in the system with cactus bugs. So this cactus bug is a really cool system because these cactus bugs, their habitat is restricted to these very discrete cactus patches that can be mapped out and modeled and manipulated to assess connectivity. So we can ask, for example, is a genetic structure of a connected patch compared to a center patch more similar to that of an unconnected patch to a center patch? We can also ask these questions at different scales using these network-based measures based on what we know about how these cactus bugs disperse. And we can compare this to different measures that ecologists and managers use for connectivity, like least cost path or circuit theory that incorporates these redundancies and paths that things can move, and matrix resistance between habitat patches. And we can readily quantify these things like matrix resistance out there by measuring vegetation structure in between patches. So you might imagine if there's a thick brush between patches, then cactus bugs might not be able to perceive the next patch or move through that matrix. So these are all things that we can quantify, including patch quality by counting pads of the cactus and the size of it. So in the second part of the study, we're going to be looking at recolonization. So pretend that these little red dots are occupied patches by cactus bugs. So we can go through and we can remove cactus bugs from everywhere except for that center patch, and then we can monitor how recolonization happens. So do we see individuals dispersing across that corridor? And we can ask eco-evolutionary questions about those colonists. Are they a genetic bottleneck of this original population? Do they have genes and traits associated with dispersal? So fingers crossed that this project gets funded. So in the next part of the talk, we're going to be talking about fragmentation effects on food webs and species niches. This is work from the wog-wog habitat fragmentation experiment. The wog-wog habitat fragmentation experiment, I'll just call it wog-wog because the name's too long, is set in a part of Australia that was historically dominated by eucalypt forests. Eucalypt forests are home to some really marvelous wildlife. So when we're out at wog-wog, we might see all these animals, wallabies and wombats, gliders and kookaburras. And in fact, when you go to wog-wog, you don't need to worry about an alarm clock, because every morning the kookaburras at the crack of dawn sound like this. Love that sound. But sadly, these forests have suffered from and continue to suffer from habitat loss and fragmentation. The World Wildlife Fund actually considers forests in eastern Australia to be one of the gravest deforestation fronts in the world. And if we consider all the plant and animal species in Australia that are threatened or endangered, most of them have habitat loss and fragmentation listed as one of their threats. So I'm going to give a little bit of background and history about the wog-wog experiment. Wog-wog was created by Mike Austin, Chris Margules, and Nick Nichols of CSIRO Australia in the mid-1980s, when a lot of clear cutting was going on in Australia. And plans to clear cut a forest presented an opportunity to create this experiment by leaving behind fragments. So before any clear cutting happened, they went out and they delineated this experiment, 6 blocks, and within each block, a small, medium, and large fragment, and as well as sampling in the matrix between blocks. And then they put in these pitfall trapping sites, 8 sites per fragment, 2 traps per site, stratified by distance to edge, and stratified by the underlying topography. And so they designed this experiment, they laid it out, and they collected data with these pitfall traps for invertebrates over 2 years before any manipulation happened. And then in 1987, the clear cutting began, so those eucalypt trees were harvested, and the remaining slash was burned. There's some pictures. And this clearing happened around 4 of those 6 blocks. And 2 of those blocks are located within a national park, and they serve as a reference control blocks. And so it looked something like this after they were done. So a clear-cut matrix with those eucalypt fragments left standing. And in that cleared matrix, they put in a pine plantation monoculture of Monterey pine. And if you look closely here, you can kind of make out the lines of where those Monterey pines went in. So data collection with these pitfall traps continued 4 times a year until the year 2000, when CSIRO cut funding for this. And so no data were collected between 2000 and 2009. And then in 2009, my collaborator, Kendi Davies, received funding from NSF to continue this monitoring to see what happened with the beetles that were originally collected to understand the long-term implications of fragmentation and the predictors of extinction risk. By now, those baby pines that were put in are now a mature pine plantations, so they're actually fragments of eucalypt surrounded by mature forest of mature pine plantation. So this brings us to where we are today. So, but soon, those timber companies are going to want to return on their investment on those baby pines that they put in, and they're going in and they're going to clear cut that pine plantation again, once again leaving those fragments in a cleared matrix. So I'll come back to this idea here. The niche concept can be a useful way of describing and predicting ecological responses to environmental stressors like fragmentation. Hutchinson first formalized the concept of the niche as an n-dimensional hypervolume whose axes represent the environmental conditions and resource requirements for a given species. So in his great little illustration here, we see these squirrels suspended in this floating box. That's their niche, that's constrained by these three niche axes, which are the temperature range which they like, the branch density that they like, and the size of food that they like to eat. But niches can be difficult to quantify because of the amount of data required and the challenge of determining which data are necessary for a given species. The analysis of stable isotopes provides a lens through which to view niches and to quantify these environmental conditions and resource axes integrated over space and time. So for example, the ratio of nitrogen 15 to 14 can be a resource axis that tells us something about the trophic position of a species, and we can compare the range of trophic positions as a niche axis. Plants, the way that it works is that N15 is enriched with trophic transfers. So if we consider a plant, it's low in N15. An herbivore that eats that plant is enriched relative to the plant, and a predator that eats the herbivore is enriched more. And then we can compare individuals to get an idea of the range of trophic position. But these stable isotope values of the individuals also tell us information about where the food web structure, where that individual grew. So this might seem trivial in this example here because we have very simple food webs, but in nature we often have very complex food webs that are difficult to resolve. So having this information about trophic structure from consumers, from predators, can be really useful. The ratio of carbon 13 to 12 can be a resource axis that can tell us about differences in basal sources like C3 and C4 plants. But it can also be an environmental axis and tell us something about the light and temperature and moisture conditions of an environment. So the way that this works is if we consider these two food webs here, one where it's cool and shady and moist, we see values that are more depleted. The way that works is plants at the base of the food web can afford to have their stomata open more because they don't need to worry about water loss as much, and they can discriminate those lighter isotopes out of the CO2 in the air compared to somewhere where it's hot and dry. And these niches can be combined to create what we'll call the isotopic niche. So the niche occupied by multiple individuals can tell us about, can be compared to those of another set of individuals and can tell us, perhaps tell us something about the food web structure or the environment where they live, and tell us something about an environmental stressor like fragmentation. And there are ways to get metrics of these niches that are robust to outliers and differences in sample sizes. So we can know about niche shifts that happen with fragmentation, but in the case of a generalist predator, we not only know about the niche shifts of that species, but they can tell us something about what's going on in the food web where they live and the environments where they come from. So the generalist predator that's a focal species for this study is the pale-flecked garden sun skink. It's a habitat generalist. At Wog-Wog, it occurs in that matrix, it occurs in the fragments, and it occurs in that continuous eucalypt forest. They were captured as accidental bycatch in those pitfall traps, so there's museum specimens to work with. And most importantly, they are dietary generalists. So they eat whatever invertebrates are out there that they can fit in their mouth. And this makes them good indicators of what's going on in the invertebrate food web and how it's changing with fragmentation. So I went out to the Australian National Wildlife Collection, where all these skinks are sitting in these big jars waiting for the mysteries of their tissues to be unraveled. Went out there and I collected muscle tissue from them and livers, and also their gut contents to be able to analyze prey in the guts. So analyze the muscle and liver for nitrogen and carbon stable isotope ratios, and identified the prey items with the help of our lab entomologist Andy Hicks. And so this is a picture of the prey items. So you can see, for example, here a tortoiseshell beetle and some ants and different insect parts. So the questions in this part of the talk are, how does habitat loss and fragmentation affect N15, carbon-13, and the isotopic niches that are made up of those two axes? So what should we expect to find? Well, theory often applied to fragmentation tells us that we should expect to find a collapse in trophic structure with fragmentation, such that species that are of higher trophic levels should suffer declines or extinctions preferentially over species of lower trophic levels. And so if this is going on in the invertebrate food web, and it's being represented by these skinks, we should expect the values to be highest in that continuous eucalypt forest, lower in the matrix, lower in the fragments, and lowest in the matrix. With carbon-13, Australia has these great gradients that go from entirely C4 to entirely C3 with everything in between. This site has entirely C3 plants, but we might expect to see shifts with different environmental conditions, abiotic conditions associated with the fragmentation treatments. And for the isotopic niches, we might expect that fragmentation might restrict the variety of prey items, and if that variety is reflected in this isotopic space, we might expect contractions of those niches with fragmentation. So this is what we actually find. So there's a lot here, so I'll break it down. So starting with the carbon-13, we see a pattern where no difference in the eucalypt or the fragments, but values in the matrix are much more depleted. And this is explained by a treatment by time interaction, time since fragmentation. So I mentioned that matrix regenerated. So as that matrix regenerated from a hot, dry, cleared matrix to a forest, the values became more depleted. And the time since fragmentation in this matrix niche is reflected by these bubbles here. And so you see the bigger bubbles out here in the more depleted area. So what about for nitrogen-15? We found this cool pattern that's actually in the opposite direction of what I expected to find. So to understand what was going on here, I went and looked at the prey items, and the big difference was in these terrestrial amphipods. So these are detritivores. They're low on the food chain. They're quite common in the continuous eucalypt forest. They depend on leaf litter. And then fragmentation seems to affect their habitat, and they become more rare in the fragments. And in the matrix, the shrimp is off the menu because they don't occur there at all. And so with this low trophic item being rare, they switch to other higher trophic level prey items like spiders and ants. So it's an effect that was affected by not the spatial effect of fragmentation as predicted by theory, but by the abiotic effects on the habitat of this prey item. So these amphipods are are really cool. So that this was supported by the pitfall data, which these two REU students were interested in working out. But actually, over the long term, as that matrix regenerates and builds up its own leaf litter, the abundances of those amphipods eventually equalize. We don't know what happens to the skinks because we don't have specimens that analyzed that go back that far. These amphipods are also really interesting in that a spin-off project that came out of this is I started finding these parasitic nematodes in these skinks that were affected by fragmentation. So it turns out that this amphipod is an intermediate host for that parasitic nematode that infects the skinks and affects the transmission to the skinks. So this was an unnamed species, so when Hugh asked me what I wanted to name it, I said, of course, wogwogensis after the experiment. And I'm co-advising a graduate student with Kendi Davies, Matt Bitters, who's interested in doing some more work with skinks and parasites in this system. So, what are some takeaways from this part of the talk? Well, stable isotopes are useful for characterizing species niches. Stable isotopes of generalist predators can help us understand the effects of fragmentation that might not be very obvious. So basically, what's going under the hood. These generalist predators can be indicators for that. The theory on which I based my predictions turned out to be opposite of what I expected. So theory in fragmentation often emphasizes spatial effects of fragmentation, but ignores abiotic effects, or treats the matrix like an inhospitable matrix, like islands surrounded by ocean. And increasingly, we're learning about how important the matrix is. Work by John Vandermeer and Yvette Perfecto show how important the matrix is, and so there's a need for these effects to be better incorporated into a lot of the theory that's applied to fragmentation. We're putting together a special issue on habitat fragmentation that's due to come out in Ecology in a couple of months, and I'm co-authoring a paper on that, on these conceptual models of landscapes in the matrix, and when we should be able to afford to gloss over these details and take these binary looks at habitat and non-habitat, and when we need to use more of a continuum model of landscapes. Another paper that I'm leading in this fragmentation special issue is on fragmentation experiments and how they can provide important insights for conservation practice. So some ongoing and future work at WOGWOG. We're going to be looking at isotopic niches in beetles. So there's a diverse beetle assemblage out there. And we're going to be asking questions like, do these isotopic niches segregate among species, or maybe among trophic guilds? How do these species niches change with fragmentation? How does the overall community niche space change with fragmentation? So we can leverage this long-term data set on beetles and ask the question, are niches good predictors of extinction risk? So you might imagine a species that has a very broad isotopic niche might be able to consume different prey items and switch between different prey items that might be affected by fragmentation. We're interested in adding other niche axes to this, like things like thermal tolerances of species. And we're interested to see what happens when that matrix gets cleared again. So we're starting to collect these data for before that matrix gets cleared. Once it gets cleared, we're going to resample and we're going to do these analyses again. So in the last part of my talk, I want to talk a little bit about temporal dynamics and drivers of stability in plant pollinator networks. So pollination is one of the best studied types of interactions in ecology and evolutionary biology. And while these interactions were traditionally studied in a pairwise fashion, increasingly networks have been used to quantify the community context in which these interactions occur. A useful way to visualize these networks is in a nested matrix organization where we have plants sorted top to bottom by their generalization, while simultaneously sorting pollinators left to right by their generalization, such that we have interactions packed into this top left corner, which I'll call the core of the nested network, which represents generalists interacting with generalists, and this specialists interacting with generalists. And we tend to not find as many specialists interacting with specialists. So networks are typically presented and analyzed as compiled over multiple time steps, but decomposing these networks into their component, into their temporal components can give us insights into the ecology and evolutionary biology of these systems. The previous studies have shown that interactions are dynamic over both within season and among seasons. And so the question I want to pose here is which interactions are stable across time. So this question is important for understanding the stability of these systems under stressors like climate change and invasive species and habitat degradation. And it can also tell us about the evolution in these systems. So to address this question, I'll present two case studies of quantitative plant pollinator networks. One is a 6-year study that I do in collaboration with Natasha Chakov and Diego Vasquez in Mendoza in Argentina in the Monte Desert, where we look at annual variation across a 6-year data set. And the other one is a within-season study from Colorado, from the Mountain Research Station in a subalpine meadow. So jumping right into the punchline here. So this is the Argentina data set, and we have the shade of color on a heat ramp representing the number of years that interaction was occurred. So we first of all see that most interactions are observed in a single year. Less than a quarter of them are seen for 3 years or longer. And if we look where those occur in the nested matrix, we see that they're all close to that core of the nested matrix. So they're more stable in this nested matrix, not only in their presence-absence of occurrence, but also in the stability and the frequency of interactions. These interactions also tend to be the ones that are more frequent. So within season, in Colorado, we actually see the same pattern for stability within season, or for phenophase. Interactions that are frequent and close to the network core tend to occur, tend to be more stable. So take-homes from this part of the talk: interactions that are frequent close to the network core tend to be more stable. This suggests that these interactions have a key role in the functioning and stability of these networks. And it also suggests that the stability of this core likely drives a lot of evolutionary change in these systems. Often when we think about coevolution, we think about very specialized interactions, but that might be the exception rather than the rule, because we have this tight ecological coupling among generalists and specialist species that likely just hang on for the ride. So some generalist conclusions from this part of the talk. These fragmentation experiments can give us new insights into the ecology of fragmented landscapes, things like how species traits and landscape structure affect invasion, and how species niches can help us understand the effects of fragmentation on food webs and the environment. And it shows that the importance of time in mediating these drivers of ecological change. So I'd like to thank my collaborators, everybody who helped out in the field and the lab, and funding, and I'll take any questions.
No, three people need to leave, so we'll give them a chance, and then we'll take your questions.
Great.
On the Colorada study, there was a difference in species richness between the polygyne and the monogyne areas, right?
Yeah.
Can you say something about the nature of the ant communities that remain there?
There are, yeah, so they displace a lot of species. There are some species that appear to hang on better than others.
Is there different qualitative, sort of the qualitative difference of which species hang on and which one didn't hang on?
You know, recently I looked back to see, because I was wondering if the Nylandaria that detoxify the ant venom might be, not Nylandaria fulva, the invasive, but there's another one, Vivigula, that lives out there. And some recent work showed that they have this capacity to detoxify ant venom. And there's this other species, other invasive species that uses this as a mechanism to actually outcompete fire ants. And they are out there. But I just thought of it recently and haven't looked at it. So they do occur in great numbers, even in places where fire ants are present. So I wonder if that might be something. The other one that occurs commonly is a dory myrmex species that's really good at grabbing food and running away. So maybe that's how they put up with the fire ant densities. No.
Interested in using these two different isotopes to define this range. Is that the size, the breadth of that niche, is that defined, that's defined by variation among individuals?
Yes. So each dot is one individual.
Is there any way to, from those data, to get an idea of the plasticity of single individuals and how flexible they are? Because niche breadth could be both of those. I can't think of one, but I'm wondering if you have.
The only way I could think of it is looking at different tissues that have different turnover times. So maybe over different times you can get that information. The only other tissue I had was liver, which I think has a shorter turnover time. But the answer was the same when I looked at liver as it was when I looked at muscle. So I don't have those data to get at that, no. Yep.
For the Australian experiment, when you explained the experimental setup, there were fragments with different sizes. The other results you show us were just fragmented matrix or core. I was wondering, have you found any differences among sizes?
Yeah, there was no effective size. So if you take that fragmentation dot and explode it into 3, it's the same. Also did the analysis for edge and interior. Those relationships, both for carbon-13 and nitrogen-15, are then in the direction that you would expect, and that the individuals that are at the edge resemble more the matrix, and the individuals that are at the core resemble more the continuous forest.
So then size should matter, because you are going to have more edge.
Yeah.
In a small one.
Yeah, I would expect to find that, but I didn't.
So you mentioned at the beginning how that corridors are controversial for a whole variety of different reasons.
Uh-huh.
And the system you showed us actually was like an inverse corridor, where you were moving the natural habitat, and you're looking at the movement of an invasive species. So let's flip back to the role of corridors in terms of endemic biotas.
Mm-hmm.
Do they work? Under what conditions?
Yeah, so first of all, on the design of the experiment, so those traditionally the forests out there were longleaf pine savannas, which are much more open. So arguably the patches resemble the native habitat more than a pine plantation. So and then I guess to test the idea, all you to test the idea, you perhaps only need contrasting habitats. Having it be more applicable to conservation goals is great too. And then the second part of the question is do corridors work? So we have a lot of evidence that corridors work for a wide variety of taxa, especially for movement, but also for gene flow. And in the case of our experiment, in the corridor experiment, we actually have 20% higher species richness of plants in the connected patches than the unconnected patches. So there's a lot of evidence that corridors do work and do what we want them to. Yeah.
I think I missed something at the end there, but you were talking about the pollinator networks. Because I think you built the core based on whether it occurred year after, or how many years it occurred to make the core, right? That was the frequency. The red dots occurred more years or more time points. And then you said that the core was more stable. So I must have missed something.
Yeah, no, it's not created based on the number of times that cell occurs, but rather on the interaction. So they're just sorted top to bottom as a binary matrix simultaneous. They're nested as a binary matrix.
Together, but didn't you say that you measured the number of interactions, the number of times that interaction occurred over time was your measure of how frequent the interaction occurred?
The measure of stability was in two ways. One of them was the number of years it occurred, and also the frequency of the interaction, which we measured as the inverse of the coefficient of variation.
So there was no temporal component in developing the nested matrix.
Exactly. Yeah. Yeah. That's just based on the partners basically. Yeah.
With those pollinator networks, do you have any information on the spatial extent of those networks?
Yeah.
Any ideas about how spatially extensive they would be?
Yeah, so we measured in a couple different ways. In the Argentina data set, it's measured at two sites that I don't remember how many hectares it was, but a few. In the Colorado data set, I did transects that I measured for 15 minutes. So I have 6 transects that are 30 meters long and 2 meters wide. Yeah, but there could, sampling more space, I'm sure we'd find more heterogeneity. In the Colorado data set, the reason I did it in those samples is that we can see how the effect of sampling greater area affects whatever pattern I'm interested in. So we can get an idea of that. Yeah. Whereas in the Argentina data set, it's based on censuses. So sit at a plant for 5 minutes and watch it and record everything that came to it.
Thank you very much for